Okay, everyone. Thank you for coming. Delighted today to have our annual Richard Skalak lecture. This is a distinguished lecture in bioengineering at Columbia. I think, we had one last year, end of last year, Shu Chen, who was one of the pioneers of biomedical engineering at Columbia, and actually Richard Skalak was one of his contemporaries and is a co-pioneer. And in fact, Richard was a homegrown Columbian, got all of his degrees at Columbia, and was a professor. But he was a professor really first in civil engineering and engineering mechanics, and started off studying fluids in oil extraction. Okay, so very different from biomedical engineering. But then he teamed up with Shu Chen and started applying principles in engineering mechanics to the understanding of biomaterials, and had a very successful career, did a lot of work in terms of looking at the mechanics of the deformation of red blood cells. He did some later work in the world of dentistry. He was one of the inventors of titanium dental implants. And in fact, did something very similar to Shu Chen, which is after he built up bioengineering here. This was before the department was created, but there was an institute. And he went then with Shu Chen to the West Coast, to UCSD, where it's a little warmer, and helped create that program out there. So we always like to think of UCSD's program as Columbia West. Don't tell them that. So we're very pleased to have this year as our distinguished lecturer, Alyssa Panitch, and she's actually the chair of the Department of Biomedical Engineering at Georgia Tech and Emory. Dr. Panitch is really once again, a multidisciplinary scholar. She got her actually got 2 bachelor's degrees, one at Smith College in biochemistry, another at University of Massachusetts Amherst in chemical engineering, got her PhD at UMass, then did a postdoc in Switzerland, which is actually, I think, a nice place to do a postdoc if you can, at the University of Zurich and also ETH. She's a fellow of the Biomedical Engineering Society and AMBI, and she serves as the editor of the journal Colloids and Surfaces B, Biointerfaces. Let's welcome Dr. Panitch. Oh, yeah, sorry. And first, I would like to present the award first for the lecture. Thank you, Alyssa.
So thank you so much for the very kind introduction and the invitation to be here. It's really an honor. And I've had a great evening and morning. This is my first visit to Columbia, and I'm incredibly impressed by the collegiality, hospitality, and just the quality of the science and the faculty that I've been able to meet, and just a wonderful visit. So thank you so much. I'm going to start off just with a couple slides on my department, because I am always recruiting graduate students, postdocs, research scientists, et cetera. So for the faculty in the room, if you have any great undergraduates that you would like to recommend, please do. And for graduate students, if you're looking for postdocs afterwards, please think about us. We're a very large and very diverse faculty. So we have 19 professional faculty on the Georgia Tech campus who really focus on our undergraduate program and teaching. A couple of them also focus on our graduate program. We have 72 tenure track faculty, 48 of them at Georgia Tech, 22 at Emory, embedded in the medical school, and then 2 of us are at both Georgia Tech and at Emory. So lots of people doing lots of diverse research that might match interests. We also have several research foci or strength areas, and they're listed here. I'm not going to go through all of them, but as I said, when you have a very large faculty, you can have several strength areas. And then for people who are looking for graduate school, we also run several T32s within the department, and then there are others outside of the department also. So these are ways to fund graduate students in cohorts and communities around different disease areas. All right, so I'm going to stop there. I'm going to start off with a story that I've told a number of times. It's work that has been going on in my lab for several years and continues to go on in my lab. So you'll sort of hear the history of that. And that is in the angiogenesis area. And then I'm going to move on to some very new research that I've never talked about. So especially when I get to that story, just give me a little bit of space to describe the data because it's the first time I've presented it publicly. Okay. So I'm going to talk to you about our proteoglycan work in the context of ischemic wound healing and these open ulcers that happen due to lack of blood flow, and many of these happen in patients who are diabetic. They develop because of the lack of blood flow, but also because these patients have neuropathy, so they actually don't feel that the tissue is injured, and so they don't know that they should be offloading or that there's an injury happening, and it continues to enlarge until it's very difficult for it to heal. And it accounts for the majority of the non-traumatic lower limb amputations. And when someone has an amputation due to an ulcer, it dramatically decreases their life expectancy. So it's a huge issue for individuals. It's a really complex disease, and I'm not even going to try. We'll just use my mouse as the pointer. It's a really complex disease, and I'm going to talk to you today about two areas. One is in sort of the fibrosis that happens during the wound healing, where you get fibroblasts that become myofibroblasts, are very active and proliferative. They secrete a lot of matrix, and it causes scar formation. At the same time, they're secreting a lot of molecules that degrade the matrix, and so for people who don't heal, that degradation overwhelms the synthesis. And then from an angiogenesis perspective, you have dysfunctional endothelial cells that aren't forming new blood vessels. There's release of lots of different anti-angiogenic or anti-new blood vessel formation, and there's lots of oxidative damage that goes on. So we're going to try to address both issues throughout our solution space. So I'm going to go back in time, and I'm going to try to convince you that thinking about proteoglycans in this context is really important. So proteoglycans have a core protein, and coming off that core protein are one or more glycosaminoglycans like heparin sulfate or dermatan sulfate, chondroitin sulfate, keratin sulfate. And in the case of wound healing, proteoglycans are particularly important. There's a small leucine-rich proteoglycan called decorin that helps control collagen formation, fibril formation, as well as the degradation of collagen. And in decorin knockout mice, they have abnormal levels or abnormal collagen fibril formation, and their skin is very, very fragile. And if you look at the left side of the screen in the upper left-hand corner, there's a TEM image that comes from Renato Iozzo's group at Thomas Jefferson University. And this is skin from a wild-type mouse. And you're looking axially down the collagen fibrils, and what you'll see is they're all about the same size and pretty uniformly packed. But in mice where the decorin has been knocked out, if you look at their skin, you see the collagen fibrils are very non-uniform in diameter and not uniformly packed. And this in part leads to the fragility of the skin. It's also been found that in human fetuses, they have a high level of decorin in their skin, and they tend to heal without scarring. Young, healthy adults have a low level of decorin in their skin. They tend to scar. And healthy elderly adults, when I used to tell this story, I was not among this population, I now am, tend to take a long time to heal, but heal scar-free, or with less scar than when they were younger. So there's something about decorin that's important in wound healing and decreasing scar formation. Making these molecules is particularly difficult because you need mammalian-type post-translational modification. So you're not just making a protein. You're making a protein with a glycosaminoglycan attached at a particular point. So it's hard to use things like bacteria and yeast. And they're hard to isolate in high quantities from tissue. So my lab thought, well, what if we want to just recapitulate some of the function of decarin? But we don't need the whole molecule. What if we just took the glycosaminoglycan portion and put on a peptide that would bind to collagen? Would the fact that the glycosaminoglycan associated with collagen be enough to recapitulate the ability to control collagen fibrinogenesis and protect the collagen from degradation? Because if it can, it might help in wound healing. So indeed, this is what John Pederi did. I used to have a picture of John in this presentation. I don't know where it went. So this was his PhD work many years ago. And he took a dermatan sulfate backbone and conjugated to it about 6 collagen-binding peptides and did a ton of in vitro work to show that indeed it helps control collagen fibrinogenesis and it protects collagen from degradation by proteases. I'm going to primarily show you the in vivo work with this because this is the very beginning of the story. So to see if it actually helps prevent scarring after we did all of the in vitro characterization, we used a rat model where we put a 4-centimeter incision on the back of the rat. We then dripped a solution that had hyaluronic acid in it just to make it viscous enough so that it would stay within the wound bed. We dripped a solution of either just hyaluronic acid, just PBS, phosphate buffered saline, or hyaluronic acid with our molecule. We sutured the animal closed, and then at both 21 and 28 weeks, the data is very similar regardless of time point. We tried to look at collagen organization and scar formation. So this is the 21-day data. And what you see is in the upper left-hand corner, these are animals who've had an incision but weren't treated. They just had PBS in the wound bed. And you see the collagen fibril organization is not great, and the fibrils are different sizes. That's quantified in the histogram to the left of the image. Directly below that are animals that were treated with just HA, and again, non-uniform collagen fibrils, not well packed, and again, quantified to the left. The collagen fibrils are a little bit smaller. The top right is our decarin mimetic, which is called DS-Cili originally. Cili is for amino acids of the collagen-binding peptide. So dermatan sulfate with the collagen-binding peptide, the fibril diameter looks better, looks more uniformly packed, and if you look at the histogram, it looks very similar to the one below it, which is an uninjured section of skin. So we're starting to recapitulate the ability to control collagen fibril formation. If we look at the surface of the wound, on the left you see non-treated, and you can actually even see where the sutures went in as well as the incision line. And on the right, you can barely see any scar on the surface of the skin. Those are the treated animals. Left lower corner, again, our control, and in the red oval, you see a lot of dense granulation tissue and scar tissue where you see sort of the dense blue staining. And on the right, you see more of a basket weave of the collagen fibrils. And you see it looks much more like the area outside of the oval, which is native skin. So we see less scarring with histology. And then if we take our molecule and we put it in solution with collagen fibrils, and we treat the solution either with PBS or with MMP1, which is an enzyme from the body that degrades collagen. And then we take it and we run it on SDS Page, which is an electrophoresis method to separate protein by molecular weight. We see collagen that's just the collagen fibrils without any treatment is on the left, and you see high molecular weight bands in the gel. In the center lane, you see where the collagen fibrils were treated with the enzyme, but not our molecule, and you start to see some lower molecular weight fragments. And if we've treated the collagen with our molecule, we don't see those lower molecular weight fragments, suggesting we're protected on collagen from degradation. So with this kind of decarin mimetic, we are able to control collagen fibril formation, control the diameter of the fibrils, and start to suppress the degradation of the fibrils in response to a proteolytic enzyme. So at this point, that work was all done at Purdue. I moved to UC Davis, where I was for 6 years before moving to Georgia Tech and Emory. And I worked with Aijun Wang and Kit Lam, who are both faculty members at UC Davis. And when I got there, they had just finished panning for a peptide that would bind to the alpha V beta 3 integrin receptor. And the alpha V beta 3 integrin receptor is highly present on endothelial cells and endothelial progenitor cells. And so they were very interested in taking this peptide and grafting it to the inside of a vascular graft, so that they could put the vascular graft in an animal and hopefully capture any endothelial progenitor cells that were floating by to re-endothelialize the blood vessel. But when I saw this, I was really excited because the alpha V beta 3 integrin receptor, when it binds to cells, it actually potentiates the cell's response to VEGF, vascular endothelial cell growth factor, and it increases angiogenesis. And so the alpha V beta 3 integrin receptor interacts with the VEGF-R2 receptor to increase the VEGF-R2 receptor's activity. So we asked the question, what if we could take our Decarin mimetic, add this peptide to it, then we might have a molecule that helped organize collagen, reduce degradation within the wound bed because it was protecting the collagen, and would stimulate angiogenesis by binding through this integrin receptor. So we indeed designed that molecule, and this work was done by Tanaya Walimbi, a postdoc in my lab. And the chemistry here is unimportant, really. There are a couple ways we could make this. She uses the carboxylic acid group under matin sulfate to conjugate our peptides. What is important on this slide, so that you can follow the rest of this vignette, is how we name the molecules. So our original Decarin mimetic, which was DS-Cili, is now DS6. So that's the Decarin mimetic. If she adds, on average, 2 of the integrin binding peptides, it's now 2 LDS6. If there are 4 integrin binding peptides, it's 4 LDS6. And if there are 6, it's 6 LDS6. Okay. So she first asked the question, if she takes collagen and puts it on a surface, and then adds endothelial progenitor cells, or takes collagen, puts it on a surface, and treats it with one of the variants of the Decarin mimetic with integrin binding peptide, and then seeds endothelial cells. In this case, it's microdermal endothelial cells, or microvascular endothelial cells from skin. Does she indeed see upregulation of the VEGF-R2 receptor? So if she seeds the cells just on collagen, that's on the left, you don't see any green staining. Green staining would be the VEGF-R2 activation. With our Decarin mimetic, there's a little bit of green staining, and as you increase the number of the integrin peptides on the backbone, you increase the activation of the VEGF-R2 receptor. So this was encouraging. The next thing she asked was, if I do a tubulogenesis assay. So a tubulogenesis assay, you put endothelial cells or endothelial progenitor cells onto a matrix that they don't like to interact with. And then you give them time, and they tend to want to interact with each other and form little tubules. So she asked the question, if we put our molecule down on a collagen surface that the cells don't like to interact with, can we induce more tubule formation? It's indicative of angiogenic potential. So indeed, we did see more tubule formation. That's shown down here on the bottom picture and quantified up at the top. But perhaps more importantly, she then took these collagen gels with one of the variants and put them on a chick chorioallantoic membrane. It's a typical in vivo angiogenesis model. And you can see the chick chorioallantoic membrane up there with the red blood vessels flowing through it. She put the gels on for 24 to 36 hours and then quantified the number of new blood vessels that were growing up into the gel or around the gel. And what she finds is, if she treats with the 6 LDS6, so the variant with 6 integrin binding peptides, we see a significant increase in the angiogenesis. We see a very small increase with the Decarin mimetic itself. So with that, we move to a very complicated in vivo model. I'm only going to show you a little bit of the data from it, but I'm going to talk you through what we did. And then we did this in combination with Aijun Wang's lab. So he is in the Department of Surgery and has a great surgical team. So they created a model for us that we mimicked from the literature. They take a Zucker diabetic fatty rat. So these rats have metabolic syndrome and behave a little bit like diabetic individuals. And we create excisional wounds on the backs of the animals. And the excisional wounds that are on the outside are non-ischemic, and the excisional wounds on the inside are ischemic. They're ischemic because we put a celastic sheet between the skin and the underlying tissue. So you can't get blood vessel growth through the celastic sheet. You can only get blood vessel growth from the head or the tail. There's also incisions on the side, so no blood vessels can come from the side. The sites on the outside, you can get blood vessel growth from underneath, so they're non-ischemic. Okay. To hold our molecule, and because a lot of wound healing uses collagen scaffolds, we use small intestinal submucosa from Cook Biotech. I had a long-standing relationship with them from my time at Purdue, so they were kind enough to donate SIS to us. We could then add to the SIS either our original decorin mimetic or the decorin mimetic with six of the integrin binding peptides. And then we added endothelial progenitor cells, and we put it on the top of the wounds, okay? And we have all different variants of this, which is why I said it's complicated, but I'll talk you through the data. So first thing to show you is that if we use, in the ischemic wounds, if we use the SIS plus endothelial progenitor cells, you get some healing. If we use the SIS plus the 6 LDS6 plus endothelial progenitor cells, we get much better healing. You can see that visually on the left. And then if we just look at the non-ischemic wounds, which are quantified up on the top, what you see is the top line with the red squares is the full treatment. So it's the SIS plus the decorin mimetic with the integrin binding peptides plus endothelial progenitor cells. And you see at 14 days we get slightly better healing than if we just have SIS plus the endothelial progenitor cells, or if we have the SIS plus the original decorin mimetic plus endothelial progenitor cells. And if we look at the right, which I can't see on my screen, those are the non-ischemic wounds, but there are no endothelial progenitor cells. And again, what you see is the best treatment is the treatment with our decorin mimetic with the integrin binding peptide. It's actually a little bit more impressive in the ischemic wounds, where you see that with endothelial progenitor cells, we see a dramatic increase in the healing rate with the decorin mimetic with this particular peptide, right, with the integrin binding peptide. And if we don't have the endothelial progenitor cells, the decorin mimetic behaves just like the molecule with the integrin binding peptide. So having all three together, you get much better healing, and you can see we get about 60% closure with the full treatment and only 40% closure if we don't add the endothelial progenitor cells. Okay. We also see enhanced angiogenesis. I'm not going to take you through all of the images, but they're images of each of the different treatment types. It's quantified at the bottom, and the upshot is that in the ischemic wounds and in the non-ischemic wounds, we see improved angiogenesis if we have our molecule of interest. So it definitely is stimulating angiogenesis and in improving wound healing. Okay. So what I haven't told you is, those really ischemic wounds, I said there's a celastic sheet underneath, and there's just the skin here, and blood can only flow in from the ends, right? Those particular treatments, it's really hard to keep the endothelial progenitor cells alive. In fact, they only stay alive for 24 to 36 hours because they dry out really quickly. It's hard to keep the wound moist. So although the endothelial progenitor cells are only there for 24 to 36 hours, we see dramatic improvements in angiogenesis and wound healing 14, 21 days out. So what that told me is the cells must be secreting something during that first 24 hours that's going to the surrounding tissue and telling the surrounding cells, okay, grow into this area. So you don't need the endothelial progenitor cells there forever. You need whatever they're secreting to improve healing. So my hypothesis was that it was extracellular vesicles that were being secreted from the endothelial progenitor cells, and that was what was enhancing angiogenesis. So I have a wonderful postdoc, Vanessa Dartora, who started designing experiments to ask this question. So she went back to having a collagen network sitting on a surface, adding our molecule of interest, either the original decaron mimetic or the decaron mimetic with the 6 integrin-binding peptides, seeding endothelial progenitor cells on that, and then collecting the secretome, and either using the secretome as a whole or separating out the extracellular vesicles from the soluble fraction of the secretome and testing that on naive cells to see if they were forming tubules, and then doing proteomics to start to understand what is it that the cells are secreting, what is it from the extracellular vesicles that are stimulating angiogenesis. Okay, so what her work told us was, if we do the tubulogenesis assays, and I'll just show you the images here, was that naive cells that are subjected to the whole conditioned media from the cells that I just told you about, right, that she seeded on collagen with the molecule of interest and then collected the media, that she used that media on naive cells. And the media alone from the decaron mimetic stimulated some tubulogenesis, but for the conditioned media where the cells were stimulated with the LDS, there's dramatically more tubule formation. And if she separates out the extracellular vesicles from the soluble fraction, you see even more angiogenesis. And if you test the soluble fraction, it's dramatically reduced. So indeed, it is something in the extracellular vesicles that are secreted from these endothelial progenitor cells that are stimulating the angiogenesis and the wound healing. She then went on to do a lot of proteomics. For time, I'm not going to go through too much of the data, but she does find that control EVs and the EVs from the treated endothelial progenitor cells have some overlapping proteins and some very different proteins. She went through and used a lot of various software to start to predict what pathways should be upregulated, and the number one pathway is indeed angiogenesis. If you go through this, you see some other things that are associated with wound healing, like epidermis development. There's some upregulation of proteins involved in VEGF signaling, et cetera. So this project is ongoing. She's doing a lot of work now with one of my graduate students to start to upregulate and downregulate specific proteins in the endothelial progenitor cells in hopes of upregulating them and downregulating them in the EVs, so we can start to understand which of the proteins from the EVs are most important for stimulation of angiogenesis, and we can maybe start to engineer some of these EVs as treatments. So in conclusion, I hope that I've convinced you that this molecule LDS increases, well, I didn't show you the proliferation and migration data, but it's there and published. We increase the VEGFR2 phosphorylation and its activation. We have shown that we've been able to increase angiogenesis in vitro, and then I showed you some in vivo data too, showing increased angiogenesis. We increased the wound healing rate in these diabetic Zucker rats. And the EVs are definitely enriched in proteins that stimulate angiogenesis. So, you know, again, we're hopeful that as we continue with this, we can come up with some good therapies. All right, so people involved in this, I think I mentioned Tanaya Walimbu did most of the work. The rest of these people are folks that supported her in the lab. And then Aijun Wang and Kit Lam were my major collaborators on this, and these are folks in his lab that have also worked on this project and our funding agencies. So with that, I'm hoping that I have 15 minutes or so, that I can use to go through another project. So if everything goes right, gosh, I will tell you, oh, it didn't change up there. I have to switch share. What screen am I sharing now? 1 second, please. It did not go right. It just went right on my computer. 1 second, please. Stop share. Start sharing. Let's try this one. Yeah? Yeah. All right. So my lab has been interested in idiopathic pulmonary fibrosis, again, for a really long time. We've worked on this problem off and on for probably 15 or 20 years. And idiopathic pulmonary fibrosis is a lung fibrosis that we don't know why it happens. That's where the idiopathic portion comes from. But in the healthy lung alveoli, that's where gas exchange occurs. But when you have idiopathic pulmonary fibrosis, you start to get a fibrotic response here, and that's the brown you see on the right, that forms fibrotic tissue and it impairs gas exchange. And when someone is diagnosed with idiopathic pulmonary fibrosis, they typically have a life expectancy now of 3 to 5 years, and there are really no good treatments for this disease. So it's something that we were very interested in investigating. So around 2012, 2014, 2 drugs came on the market, nintedanib and pirfenidone. And neither is a great drug. So one is a pan-kinase inhibitor, really tyrosine kinases, but it inhibits other kinases as well. And both have a lot of off-target activities, and the off-target activities are severe enough that a lot of the IPF patients would rather just not take them and deal with the disease progression because they're so miserable when they're taking them. So they're really not great solutions. They also seem to only stop the progression of the disease rather than being able to reverse any of the disease. So we had an alternative approach, and this work started back in 2006. So we were really interested in whether we could look at the pathways a little bit differently. So at that time, pharma was targeting P38 MAP kinase, which is also not a great thing to be targeting for a whole bunch of reasons. So we thought, what if we could go downstream a little bit and target MAPCAP kinase 2, which is MK2. MK2 has been implicated both in fibrosis and inflammation. And we thought, well, if we could target this, we might get rid of the off-target activities from targeting P38. But we also didn't want to go the small molecule route for kinase inhibition because all kinases have an ATP binding site for activity, and most small molecule inhibitors target the ATP site, so it's really hard to make them specific. So we wanted to target the substrate site. So where does the kinase bind to the protein it wants to phosphorylate? So our thought was, well, let's make a peptide that mimics its binding site. So this is the molecule we designed. We were very lucky because there was some work in the literature already that had started to look at one of the particular molecules that MAPCAP kinase 2 phosphorylates, and they had started this optimization process for us. So we knew what to start with for the therapeutic region. We just had to do a bunch of optimization. I'm not going to go through the optimization. The sequence is there. To the therapeutic region, we conjugated a cell-penetrating peptide region to make a molecule that would enter cells and be able to inhibit kinase activity within the cells. So working with Ragini Vittal and David Wilkes at Indiana University, we did a mouse model of idiopathic pulmonary fibrosis. To do this, you essentially treat the mice by having them breathe in bleomycin, and this causes the mouse lungs to become very fibrotic. Mice will naturally resolve, the fibrosis will. So you have a very short window in which you can look at treatment. So we allowed the mouse lungs to become fibrotic, and then we did daily injections or nebulization of our peptide to see if we could either reverse fibrosis or prevent fibrosis. And at day 28, so this is the reversal study, and at day 28 we sacrificed the animals and looked at fibrotic tissue collagen production and activated MK2. And I don't have any of the data in here. The upshot is it's published. The upshot is we work both preventative and as a therapeutic. We do see decreased collagen production. We see decreased inflammatory cytokine production, et cetera, in the animals. And we also did a lot of work to show that MK2 is really important in human pulmonary fibrosis, and it's all published in that publication. However, when you go to treat humans, because we have a peptide, it's really hard to target that peptide to the lungs, right? You either have to make a dry powdered inhaler or a nebulized version. And when you have people with really compromised lungs, for them to be able to breathe it in deeply is a little bit problematic. And they're also a little skeptical of what are they bringing into their lungs. They don't want to make the disease worse. So if you can come up with ways that aren't dry powder inhaler or nebulization, it would be better. And so we've been thinking very recently about different ways that we could deliver drugs to the lungs. And there's been a lot of work over the last 10 years or so on extracellular vesicles, and that work is becoming much more prolific now. So we started working with Randy Carney's lab, and this is the work of Neona Lowe, whose picture I also thought was in here, but will show up at some point or was already there. I apologize. But Neona did all of this work. And in fact, she just defended last week and sent me these slides. So a big thank you to her and congratulations. But so extracellular vesicles, I mentioned them at the end of my last talk. But these vesicles are secreted by essentially every known cell. And they're lipid bilayers that encapsulate an aqueous space. And they have, within the lipid bilayer, a bunch of different proteins and glycoproteins and other molecules that are really dependent on the cell that they come from. And they're various sizes. And you can just see in this image, right, a 30 nanometer vesicle is much different in size from 150 nanometer vesicle. And these EVs can be even larger than 150 nanometers. I mean, they can be 500 nanometers. So they're very, very different. So they're very different in size. They're different in their chemical composition. And all of this depends on their biogenesis, their parent cell type, and their parent cell state. So when I say biogenesis, are they budding off from the cell membrane? Are they secreted like an exosome? So there's a lot of heterogeneity in these extracellular vesicles, but there's a lot of interest in them as well. And we'll talk about why in just a minute. But we know from the literature that you can load extracellular vesicles with different drugs, and you can do this using electroporation, simple incubation, freeze thawing. There are different ways to do it. They tend to stay within the systemic circulation for much longer than other nanoparticles, and certainly much longer than a free peptide. And they're able to cross endothelial barriers and get from the bloodstream into surrounding tissue. They also have some targeting capability. And when we started this project, everyone was quite sure that if you took endosome or extracellular vesicles from endothelial cells, they'll target back to endothelial cells, some portion of them will, as well as go to other cell types. And most certainly they're one of the body's mechanisms for drug delivery. So they deliver proximally, they deliver somewhat more distally within a tissue, and they deliver systemically. And it's because of this idea that if you isolate an extracellular vesicle, say from a diseased fibroblast from the lung, many of those will go back to the diseased fibroblasts in the lung that we started this project. Okay, and that's essentially what this is saying. So our central hypothesis is that if we took myofibroblasts or diseased fibroblasts, that we could isolate the extracellular vesicles, we could deliver them back to the diseased fibroblasts, and they would take them in and deliver a drug. And so these were Neona's aims. She first asked, can I load a cell-penetrating peptide into an EV? They go into cells, they should go into EVs. So she asked that question. She then said, if I take EVs from healthy fibroblasts, do they target back to primarily to healthy fibroblasts? And if I take them from diseased fibroblasts, do they target back to diseased fibroblasts? And then she asked the efficacy question. If I deliver MK2 to a diseased fibroblast, can I revert it to a healthy fibroblast? So one of the ways in which you characterize extracellular vesicles is by looking at proteins on the surface that are called tetraspanins. And there are three classic tetraspanins that you look at. So you can take a plate, spot different antibodies for different tetraspanins on the plate, capture the EVs, and you know any EV in a particular spot is positive for that tetraspanin. You can then use antibodies to see what other tetraspanins they contain. So Neona did that and found that very consistently when she isolated EVs from healthy cells, they always look the same with respect to these cell markers. She isolates them from disease cells, they always look the same with respect to those markers. And then she did a lot of cryo-EM. And cryo-electron microscopy is a great way to look at your total population of vesicles, but it's very low throughput. I don't know if you can see kind of the little bubble that I'm pointing out here. So that's a unilamellar extracellular vesicle. Here's a dilamellar extracellular vesicle. So she actually used cryo-EM on 3 different biological samples and a bunch of technical replicates and counted over 4,000 of these and characterized their size and shape. That data is shown on this slide. And what you'll see is over on the left of the slide, those are your circular unilamellar vesicles. And most of the EVs are these small circular unilamellar vesicles. And most of them are really small. Well, not most of them, but like a third of them are below 50 nanometers. Right, so below most of the limited detection of most of the kinds of detection that we use. So there's a whole population of EVs that we really are not studying very well, including in this study. So I just want to put that out there. There's a much smaller population that are circular and multilamellar. That's shown in the second column over. The third are non-circular and unilamellar. There are a little bit more of those than the multilamellar. And the second to last bar is the non-circular multilamellar. So there are all kinds of different morphologies that we see, but the vast majority are various sizes of the circular and unilamellar. And these are three different ways that people typically characterize EVs. So one is using NTA. This is a typical dynamic light scattering using diffusion to back out the size of the particle. And the limited detection there is really around 100 nanometers. And so you can see that we have a pretty broad distribution, but we're missing a lot of the smaller EVs. You can use an interferometer reflectance technique, and you can see smaller EVs. The limit of detection there is about 50. And you can use cryo-EM, and then we capture the even smaller EVs, but again, a really low throughput technique. I can't imagine how much time it took Neona to characterize 4,000 EVs. But you see dramatic differences, right, in the size of your EVs, but in all cases, three different biological replicates, very consistent within the technique. Okay, so one of the questions we started to ask was, can we take these EVs that we're sure are EVs, dramatically different in size, and load our cell-penetrating peptide? So we used a cell tracker that was red just to track EVs, and that just diffuses into the EVs. And then we also used varying concentrations of our MK2I, our cell-penetrating peptide, and that's shown in, I lied, the cell trace is shown in red and our peptide shown in green. And that tells you the total number of EVs and then the total amount of the EVs that are carrying the MK2 peptide. The other thing she did was add trypan blue outside the EVs, because trypan blue will quench FITC, fluorescence, and so we could determine how much of the MK2 cell-penetrating peptide was inside the EVs and how much was outside. And then we used flow. So anything that is positive for both FITC and the red tracer is in this quadrant here, so about 18% of the EVs that we could detect using flow are loaded with our peptide. Many of them, 73%, just have the tracer, and then some percentage didn't take up anything. And then if we quench so that we don't see any of the peptide on the outside of the EVs, we see that about 12% of the detectable EVs are loaded with our peptide. This is surely undercounting, right? We're not seeing the really small EVs, and if we have less of our peptide in there than the limit of detection for the flow, we're not going to see them as positive. But we know at least 12% are loaded with our peptide. What we also know is that as we increase the concentration of peptide that we incubate with the EVs, we can load more of the cell-penetrating peptide into the EVs, and this is consistent with regardless of the size of the EV. So we can get the MK2 peptide into the EV, and if you look at this shows, if you look at the purple, well, I guess it's mauve round circles, there's a solid circle and an open circle. So the top line and the second from the top line, that's the quenched and non-quenched, so it's a difference in how much is inside the EVs and how much is outside the EVs. Okay. The other thing that she found is as you increase the concentration of cell-penetrating peptide, you start to destabilize the extracellular vesicles. This isn't too surprising because the cell-penetrating peptides have to somehow destabilize the membrane to get in. If you have too much of it, you probably over-destabilize the membrane and you start to lyse the vesicles. So the work I'll show you for the last few slides is all done with this 100 micromolar concentration of cell-penetrating peptide. Okay, so I hope that we've convinced you that the MK2I loading is dose-dependent into the EVs. It affects the EV integrity, and she did this again with 3 biological replicates. It's very repeatable. So the next question was, what happens if we take EVs from healthy fibroblasts or EVs from non-healthy fibroblasts? Can we target? So to do this, she labeled native EVs from either healthy cells or disease cells with a green fluorescent dye. And then she treated either healthy fibroblasts or myofibroblasts with the EVs and said, what's taken up? So that's this data here. So if we look at the left-hand side of the graph, the first two bars are controls. So the controls are not treated or treated with a standard liposome. So not much is taken up by healthy cells. So all of the stuff on the left is healthy cells. If we treat healthy cells with a low concentration of vesicles from healthy cells, you see some uptake, that's the third bar over. You see less if it's from EVs from unhealthy cells. If we increase that concentration, you see uptake from healthy cells and a little bit less of the EVs from unhealthy cells. And at the highest concentration, which is outlined in yellow, you see good uptake from the healthy cells and slightly less uptake of EVs from unhealthy cells. So that's with our healthy cells taking up EVs. If we have disease cells taking up EVs, we see the same thing, but they take up fewer EVs. So surprisingly, if you just look at the yellow box on the right, the unhealthy cells are taking up EVs from healthy cells more than they're taking up EVs from unhealthy cells. And they're taking up way fewer EVs than the healthy counterpart. So I looked at this data, I said, oh my gosh, no wonder it's so hard to treat this disease. How do we get drugs into these unhealthy cells, right? They're just not taking stuff up. So the next set of data, I'm not going to go through in detail. It's with the MK2I loaded vesicles. We see similar things. The one thing that we do see, or two things we see, again, fewer EVs are taken up by the unhealthy cells, regardless of whether they're healthy EVs or EVs from unhealthy cells. And we get a lower amount of uptake. We also see a little bit of a bell-shaped curve, right? With the native EVs, we saw more EVs, more were uptaken. Here we see that we kind of saturate and fewer are uptaken. That's not unusual with drug dosing. So not too surprising, but means we really have to dial in the dose that we're using. Okay, so MK2I can be, our cell-penetrating peptide can be loaded into fibroblast EVs, and those EVs are taken up more than the myofibroblast EVs and more than native EVs. So if we don't load in any MK2I, the native EVs don't go in as well as the MK2I loaded EVs, and healthy cells take up the naive and the MK2I loaded EVs better than they do the EVs from our unhealthy fibroblasts. So the last piece of data I'm going to show you is from her treatment or protective test. So she took cells and she cultured them and she treated them with vesicles from either EVs from either healthy cells or disease cells that were loaded with our MK2I peptide. And then she exposed the cells to TGF-beta. So treatment first, then exposure to an inflammatory stimuli. And then 4 days later, she fixed the cells and probed them for alpha smooth muscle actin. So she asked, are they becoming myofibroblast-like? And what we see is in her control cells that are untreated with anything, she sees a high level of alpha smooth muscle actin. So they're treated with TGF-beta, but not with any vesicles. If she treats with free MK2I peptide to inhibit MK2 activation, we see a decrease in alpha smooth muscle expression. At low levels of healthy fibroblasts, we see, is that right? Yeah, at low levels of healthy fibroblasts, we see they behave just like free MK2I peptide. At higher levels of EVs from healthy fibroblasts, we see much better activity than the free peptide. And surprisingly, at high concentrations of the diseased EVs, we actually see equally as good suppression of alpha smooth muscle actin in the diseased fibroblasts. So maybe oh my goodness, we can't treat these cells was wrong. There may be something about the diseased fibroblast coupled with our MK2I peptide that is actually more preventative of fibrosis. And so at this point, we're starting to ask the question of, what's going on? What's the contents of the EVs? And then moving to patient-specific samples. I do have a therapeutic set of data as well. It also looks promising, but I have some questions about it still, so I'm not ready to present it real time. But it looks pretty impressive as well. So for the preventative takeaway, higher doses of the MK2I loaded fibroblast and myofibroblast EVs definitely demonstrate better activity than the free MK2I peptide. And the data from the treatment model looks promising. And so stay tuned. There'll be more data soon. So in conclusion, I'm very hopeful about this idea of being able to load these EVs with cell-penetrating peptides and other treatments. We do see that the EVs are taken up by both healthy and diseased fibroblasts, although not quite in the way we anticipated, but we still seem to see a good treatment effect. So with that, this was done in collaboration with Randy Carney at UC Davis, who's an expert in EVs and EV characterization. And Neona's here somewhere. There she is in the back of this picture. So this is the Carney lab. This is my lab. I'd like to thank you all so much for your attention. Neona was funded by this T32. I'd be happy to take any questions.
Sorry, I might have missed something, but why not put like cell markers or like the antigen markers of the target cell on the exosome, on the liposome?
Yeah, so essentially thinking about taking either liposomes or our EVs and putting some sort of peptide or other ligand to target the cells that we're trying to deliver to. Is that what you're asking? Yeah. So we've actually done a little bit of that as well, but we were hoping, there's a whole bunch of literature out there that suggests that if you take EVs from a particular cell type, they largely go back to that cell type. So our hypothesis going in was that if we just took native EVs from these diseased cells, that they would target back to the diseased cells, so we wouldn't need to do anything else to engineer them. We just needed to load our peptide. I think, it remains to be seen. All of our data is in vitro right now. We haven't moved in vivo. And if we don't see an effect with just the native autotrophism, then we would have to, or autotropism, we would have to do that targeting. So yeah, thank you.
Thank you. First of all, thank you for the lecture. I wanted to make sure I didn't misunderstand. When you were talking about mice models and the fibrous lungs, did you say that mice naturally recovered from fibrous lungs?
At least with this bleomycin model, they do. So you have to be really careful with your controls to make sure what you're seeing is the effect of your drug rather than just resolution of the fibrosis. So I always want to disclose that they do naturally recover. Why they recover better than humans do, I don't know. Although my sister-in-law is an urgent care physician who saw a lot of patients during COVID and sees a lot of patients with lung injury. And there are some individuals who seem to resolve lung fibrosis while most don't. So understanding why that is would be really interesting, right? Because that would allow us to go after treatments.
Okay, so still not entirely studied. Thank you.
Yeah, thanks.
Also, thank you for the lecture. I was wondering if the work from the first part of your lecture would have any application to maybe circumventing any of the problems in keloid scarring or like post-operative, maybe abnormal healing of the skin?
Yeah, so very intuitive question. We are hopeful that it will. We are very interested in keloid scarring, and so my postdoc, Vanessa D'Artoro, who's working on that right now, who's been working on this project, is trying to answer that question right now. So hopefully I'll be able to give you an answer in the not too distant future.
Thank you.
Yeah.
On that note, is it possible for the idea to compete with existing keloid therapies such as silicone gel sheets?
Yeah, so we're hoping that it will. And, we're going after that question in two different ways. So I am hopeful that if we can use these EVs that seem to improve wound healing, and we saw better closure, we saw better organization of the collagen, that it will impact keloid scarring. I'm intrigued with the second part of my talk as well with respect to keloids, because keloids really are a hypertrophic tissue, right? And that is true of the fibrosis in lungs as well. And so if we can come up with this kind of delivery system that really goes to the myofibroblasts or the activated fibroblasts, or potentially even to healthy fibroblasts to get them not to become fibrotic, then maybe we can treat keloids with this kind of MK2 loaded vesicle as well.
Thank you.
Do we have any other questions?
Alyssa, thank you so much. That was great.
Thank you all.